Guys, I'm Vedika. Today, let's start with morphology of bacteria. First, let's discuss what is microscopy. Microscopy is examination of minute objects by the means of microscope. Microscopy is divided into two portions, light microscopy and electron microscopy. Light microscopy is further divided into bright field, phase contrast, dark field, and fluorescent microscopy. Electron microscopy is divided into transmission and scanning electron microscopy. Now, morphology of bacterial cell. Shape of bacterial cell can be spherical, rod, curved, or spiral. They can be viewed from phase contrast microscope. Their diameter range from 0.2 to 15 micron meter and length from 1 to 6 micron meter. Shapes of bacterial cell: spherical or oval, can be diplococcus, chains as streptococci, clusters as staphylococci. Rods can also be known as bacilli. They can be single or in chains. Vibrios, they are curved rods. Diplococci are cluster or just pair of two cocci. Streptococci are chains and staphylococci are clusters. Bacilli are rods. Cucubacilli are short rod-like bacteria. Fusiform bacilli are spindle-like. Filamentous are thread-like. Vibrios are comma-like. And spirilla are spiral-shaped. Structure or cell surface. It mainly consists of capsule, cell wall, cell membrane and flagellum if the cell is motile, and also 6 pili in some case. Flagellum. It can be divided into three parts: basal body, which is associated with cytoplasmic membrane and cell wall; hook, which helps to project filament outside the cell body; helical filament, which helps in locomotion. Gram-negative flagella usually have L ring in the plane of lipopolysaccharide in the outer membrane, P ring in the periplasmic region, and MS ring located within or above the cytoplasmic membrane. Location of flagella varies for different species. Monotrichous extend from one end of the cell. Amphitrichous, single or cluster of flagella at both cell poles. Lophotrichous, a cluster of polar flagella from one end. And peritrichous, lateral flagella. Pili. They are hollow, non-helical, filamentous structures. They are shorter and more numerous than filament or flagella. They are non-motile but can be present in both motile and non-motile bacteria. They play a great role in attachment. 6 pili are involved in conjugation. Capsule. It is the outermost layer. The loose association with the cell wall is known as line. Coulomb reaction. In the presence of specific antibody that reacts with capsule, it becomes swollen. When capsule is absent, the bacteria is avirulent. It is surrounded by hydrophilic polysaccharide which helps in attachment with host. D-glutamic acid. Unnatural form of amino acid that protects organism from degrading effects of host proteolytic enzyme present in capsule of Bacillus anthracis. It protects against temporary drying by binding water molecule. Blocks attachment of bacteriophage. It is antiphagocytic in nature. Promote attachment of bacteria to surface and play a role in compound having an electric charge. Cell wall. It gives shape to bacteria. prevent the cell from expanding. Comprises 10 to 40% dry weight of the cell. Main role in growth and division. The shape determining part of cell is peptidoglycan, which is also called as murine. Peptidoglycan. Peptidoglycan is the polymer of N-acetylglucosamine, N-acetylmuramic acid, L-alanine, D-alanine, D-glutamate, and diamino acid. Concept of gram staining. It is the identification of bacteria based on the structure of bacterial cell wall. Bacteria are treated with crystal violet and iodine, followed by washing with alcohol or acetone, and counterstaining with a red dye, saffron. Bacteria which are decolorized by alcohol take up the red color and they are termed as gram-negative bacteria. Bacteria that retain the crystal violet stain and do not get decolorized by alcohol or acetone take up the purple color and are termed as gram-positive bacteria. Structural difference between gram-positive and gram-negative bacteria. In gram-positive, peptidoglycan amount is more than gram-negative bacteria. In addition, gram-negative bacteria has an outer membrane. Protoplast. Bacterial cell without cell wall prepared from gram-positive bacteria with treatment of lysozyme or by growing it in antibiotic. Spheroplast. When gram-negative bacteria is treated with lysozyme, peptidoglycan of the cell wall also degenerates. This generates two membranes, outer and plasma. L forms. Bacterial cell with defective cell wall and irregular growth and multiplication. They are produced by degradation of peptidoglycan by bacteriolytic enzyme or affecting its synthesis by inhibitors or by mutations essential genes for cell wall synthesis. Cytoplasmic membrane. It consists of two phospholipid layers. Has two types of protein, integral protein which is embedded in the membrane and peripheral protein which lies on the surface. Phospholipid structure has two components, hydrophilic and hydrophobic. Hydrophilic is polar and hydrophobic is non-polar. Thickness of cytoplasmic membrane is 7.5 nanometer. Cytoplasmic membrane has phospholipid 20 to 30% and protein 60 to 70%. It acts as hydrophobic barrier. Enzymes for respiratory metabolism are present on it. Lipid layer has fluidity. Mesosomes are invaginations and haploids are absent. Mesosomes. Mesosomes are folded invagination in the plasma membrane, common in gram-positive bacterial cell. Mesosomes are of two types, central and peripheral. Central. Penetrate deeply in cytoplasm, near middle of the cell, appear attached to the nuclear material. Peripheral is shallow, non-restricted and not attached to nuclear material. Mesosome play role in cell division. Cytoplasm, the main body of the cell. It contains structures like mesosome, nucleoid, ribosomes and cytoplasmic inclusions. Cytoplasm is divided into three areas: cytoplasmic area, granular in appearance, chromatic area, rich in DNA, fluid portion with dissolved substance. Ribosomes in bacterial cell are 70S, composed of 50S and 30S, while eukaryotes have 80S, which are composed of 60S and 40S. S stands for sedimentation coefficient. Nuclear material. Prokaryotic organisms have nucleoid as nuclear material, and eukaryotics have nucleolus plus nuclear membrane as their nuclear material. Prokaryotic nuclear material do not have membrane-bound nucleus. It contains bacterial chromosome. Nucleoid can be seen in light microscope by fulgurant staining, which is specific for DNA. The nuclear material is very long. In electron microscope, it is seen as light area with delicate fibrillar structure. Spores. Spores are of two types, endospores and exospores. Endospores. They are thick-walled, highly retractile bodies produced by Bacillus, Clostridium and Sporosarcina and few others. Shapes. Terminal. Present in Clostridium tetani. Subterminal. Present in Bacillus cirrus. And central, present in Bacillus subtilis. Sporulation occurs in unfavorable conditions. Formed within 8 hours. DNA gets replicated. Spore septum is formed between the DNA and rest of the cell. Plasma membrane pinches and becomes double of the membrane, which is known as forespore. Germination. Formation of vegetative cell from spores is called germination. Exospores. Methylocene genus forms exospores by budding at one end of the cell. They are heat resistant. DPA, which is dipicolinic acid, is absent and plays important role in heat resistance. Structure of endospore. Endospores are made of central spore cell which is surrounded by various layers. The outermost layer is exosporium, a thin protein covering. Below this is the spore coat, which is composed of highly cross-linked keratin and layers of spore-specific protein. The cortex consists of loosely cross-linked peptidoglycan and contains dipicolinic acid, which is particular to all the bacterial endospore. Core. Core contains the cell wall, cytoplasmic membrane, nucleoid and cytoplasm. It contains 10 to 30% of water, which contributes to gel state. SASP. Small acid soluble spore proteins. They are formed during sporulation and bind to DNA in core. They protect the DNA from UV light and dry heat. Thank you for watching this video.